Hello, everybody. It's a great pleasure to welcome you this afternoon to listen to Alexandra Hammers. The project Cortex, LabEx, and indeed Neurodis is to bring new blood to Lyon. And Alexandra Hammers is an example of that because he's had a long career in England, where he was at
UCL, then Imperial.
Right. And has been in Lyon now for 3 years, so I think you all know him very well. And he's doing wonderful things with whole brain imaging, and he's going to tell us how you can use this to find uncharted territories of the brain. Thank you.
Thanks a lot, Henry. My background is I'm a neurologist, epileptologist. I've done an MD in morphological imaging with MRI, and then a PhD in quantitative PET imaging, but I'm not going to talk about PET much today. Henry asked me to do a conceptual talk and not talk about everything I've ever done, so I've tried to conform to that. We'll be explaining the title as we go along. So I will be giving a very brief introduction because I realize many of you are neuroscientists and don't have anything to do with imaging. So we'll give a very brief overview of imaging and image analysis. Then I'll talk about 3 strategies for using digital brain atlases. I'll give some examples about extracting information from images of healthy subjects, and then talk about 2 clinical domains, epilepsy and neurodegeneration, where atlases can help to render the invisible visible, and finish with an outlook of 3 opportunities. So first of all, an introduction. When we talk about atlases, you can have atlases at all kinds of levels. We've heard Henry Kennedy's fantastic talk 2 weeks ago about hodology and connections between regions at the microscopical level. So the first level you can use in brain imaging, or indeed in clinical neurology, is the whole brain. And indeed, that level may be entirely sufficient if, for example, you take a CSF sample to determine amyloid beta or tau proteins to determine the likelihood that a person with dementia has Alzheimer's disease. That's a whole brain approach. The next level down are the lobes, and there are diseases which preferentially affect the lobes. We'll be talking about lobes again later. Then you have the level of gyri, and you can go further down to the level of cells. Here we see a brain slab from the Allen Institute. This short video is taken from Alan Jones' TED Talk from July last year. You can go into the brain, zoom in on the cells, and go even further. So we're seeing here the cells in the hippocampus, and we'll be entering the matrix and see the cells. Of course, the cells then have organelles, and for example, we can go into the nucleus of the cell. At least I hope we can. So much for trying out the videos 3 times. There we go. So in the nucleus, you've got the chromosomes, you've got the DNA. The DNA will be transcribed and translated into proteins. The proteins are, for example, the receptors we can investigate with quantitative ligand PET. And the proteins will also have interactions one with another. And then you can study the hodology, the connections, and all kinds of things. So these are the various levels of study for the brain. And today, all I'm going to talk about is this level, the gyri. This morning band here is just to remind me to mention that the brain is in a box. As long as you keep the skull intact, if you do brain imaging, normally you can transfer information from one image to another image modality. This will be an important concept later on. Now for the first part of the title, not all of you are from Lyon, but the quote is from a famous Lyon author, Antoine de Saint-Exupéry. When you've flown in or out of Lyon, you have heard the name, and his famous book, The Little Prince, Le Petit Prince. And we've just adapted this for brain imaging because a major theme of our work is to extract more information out of brain images than you can see with the naked eye. So, as promised, 3 methods very quickly. The traditional method of brain analysis is visual analysis. If you go to the doctor and have a chest x-ray, this is how it's going to be analyzed. In all likelihood, if you ever have a brain MRI, this is also how it's going to be analyzed. And this is very good because our brains are wired by evolution to detect asymmetries very quickly, and this works very well. It's quite reproducible and the basis for all clinical use of imaging. This, by definition, always happens in the individual space. You can also use regions of interest to sample a structure, to outline a structure, to get information from another type of imaging modality, and do this for many regions of interest. This is a type of analysis that can happen in individual space or stereotaxic space. What's stereotaxic space? It's a convention space, an average of many brains, which is a coordinate system which is used to allow interlab comparisons. Now, another method is statistical parametric mapping or other voxel-based techniques where you take your raw data, you spatially normalize it to conform to a template in stereotactic space. So this, by definition, is the space in which the image is analyzed. You can see that these images are now more similar than those images, but you can still see individual gyri and salsi, which we're not interested in this particular instance. So the images are then smoothed, which means that, for example, this voxel here will be in the same area in all of the patient scans and in all of the control scans, and then you can then do simple t-tests at the level of every voxel and do the appropriate corrections for multiple comparisons and so on. And by definition, this normally happens in stereotaxic space. So after this brief introduction about brain imaging in general, what about digital brain atlases? There are essentially 3 strategies. The first, the definitions. An atlas in this talk, for the purposes of this talk, is an image with labels or RIs or regions of interest. Why would you want regions of interest? You can use them for data acquisition, for example, for PET data, we'd see that later, data reduction, information extraction, and decision making, as for example, an automatic disease classification. Getting ROIs manually by outlining things on an image is the traditional way. It's the clear gold standard. It's the only way of getting the information from hundreds of years of anatomical work into your images, but it's impossible to obtain. All of you who have ever done this know that it's just terribly boring, takes a long time, and it's just too expensive and not scalable. It was possible when you had 10 images in a study. Rolf recently did a study with nearly 1,000 brains. There is no way you can ever manually outline multiple regions on 1,000 brains. So we need automatic methods. The oldest way of doing this is to say, okay, I've got an atlas. Here you see RI superimposed on an MRI. And I will take this atlas and put it onto another brain. Here is an example. You can see a brain on the left, a brain on the right, and in the middle, this loops through a multi-stage procedure which makes this brain look more like that brain. If you look at certain areas, you will see this. This is a multi-stage method. The one we use is a multi-stage method developed originally in the field of breast imaging, and the brain deforms less than a breast. It works quite well. Now, what happens if you do this? In his seminal 2006 neuroimage paper, Rolf Heckermann here in the first row has looked at all possible combinations from 30 atlases. So basically, you warp each atlas onto each other possible brain, and you measure the overlap. This is one way of measuring this. The overlap is about 0.75. What does this mean? This means it's about as good as a student whom you've trained for a day or two. So it's okay, but a student can do better. What are the reasons why you wouldn't want to use a single atlas? First of all, because it's no better than a student whom you've trained for a day. Secondly, because there are other reasons. We all have different faces. We all have different brains. This is possibly the most used brain in the history of neuroimaging, apart from the Talarak hemisphere. This is Colin Holmes' brain, who in the late 90s scanned himself 27 times. Wonderful image with very high signal to noise, but some idiosyncrasies. For example, his left occipital lobe goes over the midline and displaces this superior sagittal sinus. And he's got a normal variant for his central sulcus on the left, which is Y-shaped, which is a variant only present in 12% of people. This is quite amusing when you go to conferences where people say, oh, we superimposed our results on the individual subject's brain. And you wouldn't believe how many people have this normal variant. So there are reasons not to take a single brain. What can you do? What else can you do? Well, we happen to have 30 atlases, all totally manually drawn over the past decade, about 100,000 slices. Would I do it again? Probably not. Anyway, we've got them, so what can we do with them? In the early 2000s, we used this to warp all the MR images into a stereotaxic space, drag the atlases along, and create what we called a maximum probability map. That's to say, for each voxel, we had 30 votes, and we did something democratic. We said, okay, the label that gets the most votes in this voxel will obtain the label. So this one was labeled pre-central gyrus by the majority of atlases warped into this space. This approach has worked fairly well. And is definitely good enough for example, PET imaging with a low spatial resolution. You can see the overlap has gone up to about 0.8 if you use this method, which is similar to a student whom you've trained for about 3 weeks. So lots of people have taken up this approach, especially in the field of PET imaging, but you can do still better. So when we started collaborating with Rolf in 2004, he talked to us about an approach first used in bee brains. You can see that bee brains are a lot more complex than human brains in terms of shape, and that is the warping, the high-dimensional warping of the atlas-associated images directly to the target image, followed by the same democratic voting process directly in target space. Now, why would this be better than the maximum probability approach? Because it conserves the information contained in the manual outlines up until the last minute, and also because there will be random errors in the registration process, and there will be random uncorrelated errors in the manual outlines, and they will cancel out to a degree. So when you look at the performance, these are the blue dots mean the difference between manual and automatic labeling, and you can see that from the single atlas to the single atlas with a better registration method to the multi-atlas better registration method, the differences decrease and become fairly minor, really. So when we look at the performance on the same scale as before, we can see that now we get up to 0.83 or so. And this is, as Rolf likes to remind me, pretty much the performance of an experienced rater for most structures. So a method that can now compete with manual labeling. And this is a big red X from a talk I gave recently, making the point that in 2012 there's really no use anymore, in my opinion, for single atlases, because you can get much better with the same computational cost just by using multi-subject atlases. Okay, now information extraction in in healthy subjects. We've got this cohort of 30 manually created atlases. What can we do with them? This slide to try and convince you that just like when you wander through Rome, you will only see what you know. When you look at a brain, you will only see what you know. Who can tell me where the right and the left is? They look pretty similar, but they aren't. Okay? But if you don't know, you will never find out. So first of all, we've got the petalia, which are the most anterior and posterior parts of the brain. And the right petalia normally extend further forward than the left one further backward. This is known as the brain torque. Then the angle of the ventricles with the midline is a bit more obtuse on the right side. The right hemisphere on a slice like this is a bit more pear-shaped, following the direction of this ventricle. So this will be outlined here, so pear. Whereas the left hemisphere is more egg-shaped. And those of you who have come to the Neuroanatomy for Images course know this picture already, but normally afterwards people remember, and this helps them find out what the right and the left is. This has practical relevance because we get images from lots of collaborators now, and you can never be sure that they've labeled the right and the left correctly. And while this doesn't work in every single brain, if you've got 10 brains, normally you can tell what the right and the left is. Now, let's take this idea further. We only see what we know. We have to have knowledge to make sense of brain images. Henry Moore in the late 17th century, when the likes of Willis and Wren were dissecting corpses and looking at the brain, was very dismissive of the idea that you could that this is something you could ever think with, and he dismissively called the brain a bowl of curds or of fresh cheese. Now, if you look at it like this, yes, it's very difficult to make sense of it. But if you look at the whole picture, it becomes a lot clearer. If you've got labels to help you, all of a sudden you see the structures, you see the recurring patterns of the gyri and so on. So I think then you have to take off the label bowl of curds. Now, if we look at normal aging, for example, in this section we're talking about healthy subjects. Here you can see an evolution of a large number, about 500 brains between the ages of 20 and I believe 80 or so. You can see the change of shape, the widening of the ventricles, and so on. This is an excellent visual impression to what is happening this very moment to all of us. But how can you quantify it? So if you've got labels to quantify the regional atrophy here, this looks different. And all of a sudden, you get more out of this, because obviously the ventricles go up with age, and it's a nonlinear process, and you can put boundaries on it and so on. But when we did this for over 500 subjects from the ICSI database, we discovered other interesting stuff. For example, there is a large number of regions which decrease in volume, always relative to the total intracranial volume, with age, so a gentle decline. There are these nonlinear things, and there are other structures which do very interesting things. For example, the hippocampus actually increases in shape up until your age. I mean, the age of the beholder. So this is a phenomenon that was seen in quite a few structures, and something you simply couldn't see by just looking at the images like in the previous video. We have now, we started with healthy adults. We've now gone down and provided atlases for 2-year-olds and 1-year-olds, many of them available from this webpage or otherwise via a license directly from me. Very recently, we published neonatal atlases, manually produced. I will show later how we're now able to label the brains of very elderly subjects, and in collaboration with the Allen Institute, we've also tried to label post-mortem brains. These were their first post-mortem brains, and you can see that they hadn't been correctly stored after death, and they resemble somewhat a loaf of bread. These days, they've got much better quality images. 2 weeks ago, Henry Kennedy told us about diffusion tensor imaging and tractography, and that it's only good for producing pretty pictures. So I haven't dared putting tractography or connectivity here. I've just put diffuse connections, and the connections become all the more diffuse because this has now diffused into popular culture, and you can see DTI on the latest cover by the pop band Muse. But it's still useful for something. It doesn't tell you about directionality. It's not very fine-grained, but it can be useful. This was a collaboration with people from King's College London, where we've used quantitative T1 maps, which are great for gray-white matter contrast. We labeled them with our atlases, with the multi-atlas method I've shown earlier. And then they looked at the connections between the thalamus and all of these cortical regions. And this is a map of the connections tested 16 times. So the same subject was scanned 16 times, and the reproducibility. So in yellow, you've got areas where the same area was shown as connected 16 times, and this goes down to red, where it was only overlapping in 5 subjects, but fairly reproducible within subjects and between subjects. So I've called this knowledge propagated, because these aren't regions we have outlined. These are the regions we've outlined. We've outlined the thalamus as a whole, but the subdivisions come from the knowledge we've put in this, plus the knowledge incorporated in the DTI images, which then give information about the connectivity of thalamic subdivisions. We've also used this technique at Imperial for looking at connectivity differences during aging. These are matrices where the left and the right hemisphere are plotted. You can see that the intrahemispheric connections are more frequent than between hemispheric connections as measured with DTI. These are younger subjects, these are older subjects, these are the significant differences, and you can then use this for machine learning techniques like support vector machines and the like to automatically classify people into young and old. I don't tell you what the people said who were 25 and whose brain was classified as 65, but on the whole this works fairly well and shows that there are systematic differences across the lifespan. The next 2 slides will be a bit speculative, and in fact this is speculation which was really inspired by Henry's talk 2 weeks ago, where I thought, well, this is really distance rediscovered, the multitude of connections between neighboring gyri. And when you look at the literature, there is a clear distance effect for structural and functional connectivity. There are genetic effects. I found this paper quite by chance last night and was pleased to see that this is an ex-student of mine who has found genetic clustering which often conforms to lobes or major sulci, but not always. You can see here this frontoparietal region. And the Allen Institute has recently made available their data and published in Nature, where they used a kind of gene eigenvectors of gene expression and plotted this in MNI space and found a very good spatial correspondence. So distances in the brain do matter, which of course is a source of great pleasure for people with a neuroanatomical interest like me. And oh, there we go. And people have tried to use this for looking at correlations between regions. On the left, you see a work from Canada looking at correlations in terms of thickness. Again, the regions are plotted here and here. This is the 45-degree line, and you can see that there are some clusters. So yesterday, we thought, well, what if we try the same thing? Now, do you see any correlations here? Yes, but they aren't ordered. This is because we used random.org to create a random ordering of the regions in the atlas. And of course, there is no order in it. But when we do order them by neuroanatomical criteria, then all of a sudden these patterns appear. You can distinguish the squares in the individual lobes. And you also find clusters which overlap between lobes. So this dates from yesterday. It was inspired by you from 2 weeks ago. We would see where this leads, because you could imagine that this might be useful for distinguishing interregional correlations in various diseases, for example. Okay, away from the entirely speculative and back to something one might actually use in clinic, rendering the invisible visible in epilepsy and neurodegeneration. I do think I have been pressing on the laser button quite a bit too often. You mentioned you might have fresh batteries. I think I might come back to you on that. And I try not to use the laser too often. So applications epilepsy. Of course, it gets stuck on the most boring slide. Yeah, I think so. These are triple A's. In the meantime, it has recovered a bit. So we've used multi-atlasing methods for first detecting the visible. This is a bit boring, but it's always good to check that the methods you're using are actually showing you what they are supposed to be showing you. And what you see here in blue and red are the right and left hippocampi of controls and patients. And we see that the right-left difference, which is physiological, is, is reproduced. And we also see that the automatic method here overestimates the hippocampal sclerosis, the shrinking of the hippocampus a little bit. This is due to small misregistrations here. Is this good enough? Well, we got 100% sensitivity, which is what you need for a screening technique. And by looking at asymmetries or suitable cutoffs with ROC curves, we also got 100% specificity. Can't say fairer than that. But as I said, this is boring because this is something that an experienced epileptologist or neuroradiologist would see anyway. So can we can we go further and make the invisible visible? So here we used 83 region segmentations and data reduction techniques and machine learning to classify people with temporal lobe epilepsy and either hippocampal sclerosis or entirely normal MRI. And what we see is that compared to controls, we get a 91% accuracy rate. So the technique detects 91% of people with epilepsy correctly as people with epilepsy. This is not a clinically useful situation because we know they've got epilepsy. We want to know, does it come from the right or from the left, when their clinical semiology tells us that they probably have temporal lobe epilepsy. So that's the situation in the last row. And there we got a whopping 94% of accuracy, which I found absolutely astounding, seeing that these are images which the best neuroradiologists have not been able to classify correctly. So an extremely promising result published this year. But the story doesn't end there, because when we looked at which structures were driving the classification in the people with hippocampal sclerosis, of course it was the hippocampus and the usual suspects. In the people with normal MRIs, some of the structures were expected, frontotemporal structures connected via the uncinate fascicle. But there were other structures which were totally unexpected, and the most unsuspected structure was the substantia nigra. We went back and checked. There was no missegmentation. There was incidentally no reason why there should be, because the controls and patients had been segmented in the same way. This was very robust and also seen in the hippocampal sclerosis patients, where it didn't make it into the top 10 for classification because the hippocampal sclerosis was driving it, but it was definitely there. So when you then go back to the literature and use my favorite technique of quantitative ligand PET to look at the dopamine system in epilepsy, yes, there is ample evidence for an involvement of dopamine in a unilateral fashion in TLE, and Carolina Chumash, who might be here, has shown bilateral involvement in bilateral epilepsies. So some evidence that what our methods make visible is actually real. Okay, now a brief word about a technique from which we'll see a few examples later. If you've got a brain, segmenting it is actually now easier than most people think. What's difficult is extracting it from the MR of the entire head, which most people think of as easy, because even slight missegmentations will then propagate into the actual segmentation. So over the past year, Rolf has developed a method called PingRAM, which has been in the top 2 of the international segmentation validation engine for all of this year, and works really well. Which is then used to do the multi-atlas labeling, distinguishing gray matter and white matter. When you've got this, how do you present the data? One way we've come up with is a so-called volumetric fingerprint, where you plot the right hemisphere and the left hemisphere. You can see the horizontal lines which separate the various lobes, and then you can plot the individual patient's volumes, always corrected for total intracranial volume, in this fashion, and get an A4 page overview of what's happening in this brain without having to look at all 180 slices. A point that John Maziotta made in the workshop we organized at BioVision off last year. He said the radiologist is now paid less for looking at hundreds of slices in MRI than they used to be for looking at one chest X-ray. So it's not a sustainable economical model, and we have to have data reduction. And this is one way of doing this. For example, there is a genetic syndrome called EAST, which gives children kidney disease and epilepsy. We've collaborated with the authors to look at the cerebral volumes of these kids, and this is the mean of the controls. You can see them in faint green in the background, and you can see that on average, these kids have smaller temporal lobes, but bigger frontal lobes than the controls. So there are systematic differences in brain structure in this syndrome, which do not always have a visible correlate on neuroradiological interpretation of imaging. What's interesting as well is that the hippocampus is a prime suspect for any type of epilepsy, but it's totally normal in these kids. So you can never prove the null hypothesis, but in those kids' images we had, we did not see any evidence for hippocampal involvement. The second application I wanted to talk about is neurodegeneration, and more specifically Alzheimer's disease. This is a typical brain of somebody with Alzheimer's disease, and you see that it's very different from the healthy 20 to 55-year-olds we used for creating the atlases. So can you segment these with the usual techniques? No. Can you segment them regardless? Yes. Rolf and Shiva Kihanenejad and others have set up a method which uses tissue class segmentation, a concatenation of these images into 4D volumes with simultaneous registration to first match the huge ventricles, match them up with the small ventricles of the atlas subjects. You still get missegmentations, as for example in this totally shrunken hippocampus, and then combine this again with tissue class segmentation to get a more restricted hippocampal volume. And you can see that most of the areas are correctly segmented. Some purists pointed out recently that we've got some dura up there. This is in the pre-Pinkram days, and this is a problem that is solved now. So we can segment brains which are hugely abnormal. Is this good for something? Rolf did a nice study looking at MRIs which were presented to scientists and neuroradiologists with overlays of the atlases where the color coding is not random but carries meaning. These are actually Z-scores compared to age-matched healthy controls. So if you've got a low Z-score, this means your structure, as for example here the temporal lobe structures, are small relative to your peers, relative to age-matched controls, which means that for the same structure size you can get different Z-scores. And we can see that the healthy subject is mainly in the middle range, the stable mild cognitive impairment patient as well. The progressive MCI patient is starting to have a lot of blue areas, except for these, and these are the ventricles, which of course go up in size. The Alzheimer's disease patient is even more affected. And when we looked at whether this helped neuroradiologists and scientists interpret the images, it was quite interesting because it had helped the scientists a lot, and the neuroradiologists were split, let's say. It didn't help them so much because they're so used to looking at the raw images, and it's so complicated to get your head around what these colors mean that they didn't find it so helpful. In other words, you can develop a fantastic technique, but it's not guaranteed that it will actually help in clinical practice. Still, I think this is something to be further developed. I would like to develop it further, if only I could. I think I should stop hitting the laser button. Okay, now if you directly ask the question, okay, you've got these regions, but there are voxel-based techniques. Is this of any relevance? Can it beat voxel-based techniques? Just to very briefly say that in some situations, voxel-based techniques are equal. In some situations, they are a little bit worse. Why? Probably because you instill this additional knowledge that certain voxels belong together to a structure which is called amygdala or hippocampus or something. Because Henry's already on stage, we're getting to the end with the outlook with 3 opportunities. The first one is of course, to further improve the techniques. We've already talked about brain extraction, which is very important, but you can go further by selecting your atlases based on similarity or age. This is a paper in collaboration with Paul Algebra, highly cited. You can use more cleverer methods for determining the boundaries of your structures, like, for example, graph cuts, and a particularly elegant study called LEAP. We used the atlases to first segment brains that were structurally similar to the atlases, and then use the secondary atlases to create tertiary, quaternary, and so on atlases. This means that even the most abnormal brains could be segmented with near unchanged accuracy. Second opportunity, constrain. Constrain what? Constrain the possibility space. For example, in quantitative ligand PET, you might have 30 frames over an hour and a half, 200,000 brain voxels, and say 2,500 different values. That's 150 billion possibilities for values there. And of course, if this information is not ordered, you can't make any sense of it. If it is ordered, it's very easy to make sense of it. So you can use this, for example, in this example, to inject the knowledge that voxels in a lobe behave similarly to constrain your PET parameters. And with the data you've got, create a much better brain image here than if you don't have this information. The third opportunity is, of course, to not sit on this knowledge, but apply it both in science and in clinical work. I've said at the very beginning that the brain's in a box, so you can use this, for example, for transferring your regions onto a PET scan, and you can see that if you do this for amyloid imaging in Alzheimer's patients, you get a 2.5 times higher value than for controls. 2.5 times is huge. There is no overlap. This is very easy to quantify. When we can also exploit the volumetric fingerprints I've shown earlier. Here an example in epilepsy, where you can see on the contralateral side a cerebellar atrophy, on the ipsilateral side also a cerebellar atrophy, but a small temporal lobe overall with a particularly small hippocampus. This is all just helping clinicians, but I think in terms of science you can look at more subtle things like thalamic asymmetries. In the dementias, this is a result from yesterday, the first morphometric fingerprints in various forms of dementia. You can see that in the temporal lobe you get an atrophy which is particularly marked in frontotemporal dementias, and in the posterior regions this is much more marked in posterior cortical atrophy. Finally, in terms of application, most of you know that we'll be getting an MR PET machine in Lyon at the end of 2013. Why MR PET? Because you can look at processes, you can simultaneously look at processes with MR and PET, so this will be important for things that happen within minutes, like neurotransmitter release, and I think it's quite exciting to be able to join up information over minutes, hours, centuries of neuroanatomical research with these new technologies. To conclude, Manual RI labeling instills knowledge and bears dividends, like we've seen, for example, in the case of tractography, where you can use the knowledge you've put into the images to further to get information down the line. The labeling is application ready. We can now do this. We've worked with, I don't know, at least 30 collaborators. We can basically segment anybody's data. RIs lead to a data reduction, which can be useful in, for example, automatic disease classification. It gives you a readout on the human scale. It's a lot easier to read morphometric fingerprints than voxel-based data. The usefulness of the whole thing is, of course, dependent on the regionality of the process under study. At the very beginning, we talked about CSF probes and the amyloid or protein tau levels in there. That's the whole brain. You don't necessarily need even the lower level for that. But in some scientific and clinical applications, you do. And routine clinical use in individual patients is now achievable. There are a great many people to thank. First of all, the Atlas team, who during the past 10 years did all those thousands of regions with me. People in Lyon, London, the funders, and a great many others who are only for whom there wasn't space on the slide. And I'd like to thank you for your attention.
I should have been more careful about the pretty pictures. Questions? I'd just like to ask a question myself. You can measure cortical thickness with some accuracy using these techniques, and that should be a handle to be able to relate your atlases to actual Broadman atlases, because there are cortical thicknesses that you can see looking at histological sections. Couldn't that be a way of improving your creation of a standard that would be easily into which you can easily put a single brain?
That's certainly something we haven't looked into enough. I feel slightly vindicated about our approach so far. You remember the slide with the bluish dots on the left and the greenish dots on the right. I think we get more convincing intra-lobe correlations with our method than the other one, which was explicitly looking at cortical thickness, whereas we only looked at volumes. Having said this, this was a paper from 2007. They used a registration method which is inferior to ours. In fact, since then, they've swapped to multi-atlas registration. And we haven't looked enough into the various cortical thickness measures, but we are doing this for voxel-based techniques in epilepsy for detection of focal cortical dysplasia, and I would certainly take up your suggestion.
Peter.
Oh, I think there was somebody with the mic before.
Okay, I was just wondering, you have this very interesting situation where overfitting your model makes it better. So the more parameters you have, the more typologies of atlases you have, the better you're able to recognize a given gyrus and give it a name. Functional areas vary. You can have the primary visual area, which is twice bigger in one individual than in another, and this will somehow maybe overtake over the gyrus or something. Do you have ideas about merging also functional data?
Can you say the last bit again?
Oh, I was wondering, do you have also ideas where you merge all this structural data with functional data, like get the topology of the visual activation in the subject, and then once you recognize the Sulcian regions, identify areas?
We don't. People have done this. Oh gosh, with these weak batteries, I don't know whether I can forward fast enough to the slide I want to show. Because you mentioned the visual area specifically, and the Russian school in the 1930s did a lot of work on that. And what I like to show is, but can't, so I'll just talk about it, is that the Sulci, in my opinion, carry much more meaning than they got credit for in the mid-2000s with people in Jülich doing the quantitative Broadman mapping and so on. And the image I wanted to show is in 2007, we've mapped their maps into the same space, which happened to be Colin Holmes space, as our maps. And on average, they are identical. So I think if you do group studies, even if you rely on morphological landmarks, you will often be fine. Your point is, of course, well taken, and the primary visual cortex is the most variable primary area I'm aware of. We would love to do collaborations with people who do visual mapping, for example. But Henry, you mentioned the Broadman areas, and can you get to them with mere thickness measures? I'm not convinced you can do that even at 3 or 7T. There are the first papers appearing from Bob Turner's lab in Leipzig and others looking at myelin mapping or ratios of one MR parameter over another. It's very interesting, but I think it's not quite there yet.
Yeah, I was interested in your thalamocortical study that you talked about in about a third of the way through. So if I understood correctly, you use your automatic methods to segment cortex and then examine using DTI the thalamocortical projections. And so did you, what I didn't see was, is there some notion of topography that you can identify there? So do you have some sort of, as we would expect from the monkey, the cortical topography that's preserved in the thalamus? And then if so, and we mentioned this to you before, it would be interesting to know if you could apply this to the corticostriatal system and so see this kind of beautiful preservation of cortical topography in the projection into the striatum.
I didn't show the topography. If I only talk about this and have more time, I tend to show the original study on this by Tim Behrens from Oxford in 2003 in Nature Neuroscience, where they had fewer regions, and you can clearly see that the frontal cortex is preliminarily connected with the anterior parts of the thalamus as you would expect it, and so on. You said thalamocortical connections. Of course, they could be corticothalamic. As we discussed before, I think the striatal subdivisions would be an obvious thing to do. Again, if you've got DTI data and reasonable T1 images, we can certainly help you set this up.
I just wanted to know, a little bit earlier you showed data showing that you've got 94% of accuracy in demonstrating the temporal yes, of the epilepsy. So that leaves with 6% of error, and I'm wondering, are those false positive or false negative or combined together?
Oh, that is a very good question. I don't know. The problem with, this was data from London, where epilepsy surgery is much less aggressive than in France or Italy. So I think most of the people with normal MRIs and temporal lobe epilepsy have not been operated upon. Because the final proof for what you're saying is operate on the lobe in question and see whether people become seizure-free or not. So these people with normal MRIs and temporal lobe epilepsy, some of them will have temporal insular epilepsy, for example, and might not even become seizure-free if you perform temporal lobe surgery. And I don't have invasive proof of laterality in those. So the 6% might just be misdiagnoses. It might be false positives, it might be false negatives. I can't tell you. But the sheer fact that these images were looked at by 2 neuroradiologists with probably a combined experience of about 60 years. And they said these MRIs are normal. And when we look at them, we can't see anything. And when you let the machine learning loose on them, based on the Atlas separations, you get 94%. So I find that still quite impressive, whatever the 6% mean.
have you ever thought of doing any kind of experimental work on animals, for example, using primage, where you would set out to make correlations between what you can see in the imaging and what you could see with pristine histological analysis? For example, if you were to try and use either functional imaging and maybe cortical thickness or histological markers of some kind to make an improved correlation that you can't do, the sort of things you can't do with human beings. I'd imagine it might not be ethical. Is that at all something that people like you envisage to do? Is that do you have any interest in primage in that sort of direction? Is that?
I have an interest. I hadn't envisaged doing these experiments myself, but if you were interested, I would love to be involved in these kinds of things. And you're right, most people do not sign the informed consent form when it says donate your brain after the experiment.
Right.
But incidentally, when you talked about the DTI visible connections between thalamus and cortex, most of them make sense. Not all of them make sense. So we know that DTI does not always reflect the real connectivity. So even with primage experiments, I wouldn't expect there to be a one-to-one mapping.
Right.
Between histology and what imaging shows.
Okay, well, thank you very much. We'll break now.
Thank you.